Now we move on to the investigation and planning section, which is the largest part of the ATP paper and usually carries around 20 marks. In this section, you will be asked to design an experiment, so it is very important to answer everything in the correct order and always remember to include appropriate safety measures where necessary. Every investigation follows a fixed sequence. First, state the aim, explaining what you want to investigate. Next, write the hypothesis, which is your prediction. Then identify the independent variable, making sure you change only one factor. After that, state the dependent variable and explain exactly how it will be measured. Next, list at least 2 control variables, such as temperature, time, pH, volume, age, species, light, or distance, which must all be kept constant to ensure a fair test. Note that, for O-Level Biology ATP, you do not need to worry about hypothesis, independent variables, dependent variables, or control variables, as these are not asked in your examination. They are mentioned only for your general understanding. Then write the apparatus required, followed by the method in clear, logical, numbered steps. Include a control experiment where the independent variable is not applied, then explain that the experiment should be repeated several times and the mean should be calculated. After that, record the results in a suitable table or graph, and finally write the conclusion, stating the relationship between the variables. This is known as the scientific method, which is the standard process used to plan and carry out scientific investigations. It begins with identifying the aim and making a hypothesis, followed by selecting the variables, listing the apparatus, writing the method, including a control, repeating the investigation for reliability, recording the results, and finally drawing a conclusion based on the evidence collected. If you think back to the practical investigations we covered throughout the O-Level Biology syllabus, you will notice that we followed these same steps every time. So, while preparing for your ATP paper, it is a good idea to revise all the practical investigations from the syllabus because they already demonstrate this complete scientific method. Your experiment should always aim to be both reliable and accurate. To improve reliability, repeat the experiment, use a larger sample size, calculate the mean of repeated results, and use organisms of the same age and species. To improve accuracy, use better and more precise equipment, such as a colorimeter, a digital balance, a thermostatically controlled water bath, and other accurate measuring instruments. Sometimes the examiner will ask how the investigation could be improved if the results were not accurate or reliable. In those questions, make sure you use appropriate improvements such as repeating the experiment, increasing the sample size, using a colorimeter, using a thermostatically controlled water bath, using a digital balance, taking measurements more frequently, controlling temperature, controlling light, controlling pH, using the same species, and using calibrated apparatus. These are common marking scheme points, so remembering these terms can help you secure easy marks.